The main topic of this video is going to be the scientific method. What is it and why is it important? And it's very important because if you understand nothing else about biology or any science at all, understand this. The scientific method is the one tool all scientists have to use. If you don't understand this tool and if you don't use this tool, then you cannot call yourself a scientist and nothing that you do can be called science. But how does this affect you if you're not a scientist? What if you're not going to be a scientist? In that case, who cares, right? Well, hopefully in this video, I'll be able to explain why it matters to you, whether you're going to be a scientist or not. So the definition of the scientific method is that it is a method by which scientists ask a question, formulate a hypothesis, and then collect data to prove or disprove their hypothesis. This is the way research is conducted in all the sciences. So how can the scientific method be used if you're not a scientist and you're not really doing what you might think of as science experiments? Well, think about it like this. Have you ever solved a problem or achieved some difficult goal, but when asked, how did you do that, you weren't really sure why? Or maybe when you try it again, you just couldn't do it a second time and just couldn't figure out why you couldn't do it again. Well, using the scientific method can help prevent situations like this from happening. So let's take a look at some examples. Let's say you're at work and your boss is trying to figure out the solution to a problem, and he goes to his employees. Who has the best solution? How can you convince him that your idea is better than the other guy's? Let's say you're using some kind of computer, whether it's your smartphone, a tablet, a laptop, and it keeps crashing. Why is it crashing? Is it an app? Is there something wrong with the computer? You could go and pay someone to figure it out for you, or maybe you could use the scientific method to solve it for yourself. Or maybe you've got a hot date this weekend and you've got this idea that you are going to impress that date with the most awesome, most excellent, most amazing souffle, but every time you make a souffle, it doesn't rise. It comes out this ugly, eggy mess. How can you use the scientific method to solve problems like these? Well, it all starts out with an observation. We'll use the last example, the souffle that keeps failing. The observation is that your souffle keeps failing. So you have a question. Why does my souffle fail to rise? Let's say you read somewhere that before you use the eggs in the souffle, they need to be room temperature and not cold. So you come up with a hypothesis. If I use eggs to make a souffle that I brought to room temperature before using them in the souffle, then the souffle I make with room temperature eggs will rise better than a souffle that uses cold eggs. So then you make a prediction. Do you think the hypothesis that you made is going to be proven true, or do you think it will be proven false? Then you conduct an experiment. Make two souffles, one using cold eggs straight from the fridge, and one with eggs that you let sit on the counter until they match room temperature. Then you check the results. What was the result? Your answer will determine the next step. Did one of your souffles rise and come out the way you wanted to? If not, then you might have to reject the hypothesis. It didn't actually work. Or maybe it did work. Your souffle that you made with room temperature eggs did rise, in which case it's possible you have a theory. But in either situation, you have to prove that there wasn't something else that caused the souffle to rise or to fail. So you have to test again. Now this is obvious if you got a failed result. You might need to change the hypothesis. You might need to retest the hypothesis to see if maybe there is some other factor. But what if the result you got from the experiment was that your hypothesis is true and you did make a successful souffle? Well, if you're going to convince others that you have the best plan for making a souffle, you're going to have to retest it. The more you test it and the more you get the same result over and over again, the stronger your evidence is that you have the best answer. But let's say you test it again, maybe you test it another 5, 10 times and you keep getting failed results, even though your first result came out right, because on retesting you kept getting a failed result, then you will have saved yourself some embarrassment of making a failed souffle that you just for sure thought, based on the first results, was going to work. Now, in order to use the scientific method accurately, there's a number of things that you have to do, and you have to do them the same way other scientists would. So that way, when you show your results, people will believe you. One of the things that you have to do is make sure you are using a good hypothesis. A good hypothesis answers a scientific question in a way that is very specific and is testable. So for example, let's look at the souffle hypothesis. If I use eggs to make a souffle that I brought to room temperature before using them, it is specific because you can tell exactly what I'm trying to test, and you can tell exactly how I'm going to test it. But there's another part to my hypothesis. Then the souffle I make with room temperature eggs will rise better than a souffle that uses cold eggs. That's testable. I just made my hypothesis testable by giving you a way to tell if it fails or succeeds. I also mentioned earlier that if your hypothesis seems to be true based on the results, you might have a theory. Might. The only way you know for sure that you have a theory is that 1, it's supported by evidence, which is what you're gathering in your experiments by repeating them over and over again, and 2, it then has to be widely accepted by the scientific community. If you get 1 or 2 scientists to say, yeah, I agree with you, not a theory. But if you can convince hundreds or thousands or millions of scientists to accept that your hypothesis is true, then and only then can you say that you have a theory. And even then, theories, no matter how widely accepted, always have the possibility of being disproven. Albert Einstein, quite possibly the most famous scientist of all time, once said, "Many experiments may prove me right." That's a theory. But it takes only 1 to prove me wrong. Because even if you have a theory, there are going to be scientists that are going to test that theory on their own. And if enough of them start to test your theory and find that it's untrue, and they can convince a number of other scientists to also accept that it's true, well, they now have a new theory that might disprove yours. Now, in addition to making sure that you have a good hypothesis, you must also make sure that the experiment you design meets certain minimum standards. One, of course, is having a good hypothesis, one that's specific and testable. 2, make sure you have clearly identified that you have a control group and an experimental group. The control group is the group for which conditions do not change, and that is the souffle made with cold eggs, because that's how I always made them. That's the normal way I've been doing. That's been failing me. The experimental group is the group that receives the variable being tested. Only one variable should be tested at a time. In this case, the experimental group is the souffle made with room temperature eggs. That's the group that's getting this new variable, this new thing being done to it. And finally, you have to make clear what your variables even are. And there's going to be 2 types of variables in any good experiment. One is the independent variable. That's what you are changing in the experiment. In this case, the temperature of the eggs before they're used is the independent variable. Your dependent variable is what you are measuring. In this case, how well the souffle rises. Your variables are things that can change either before or after an experiment. So you can think of the independent variable as the variable that you are directly changing, and your dependent variable is all the potential results for your experiment.
Make me lose with the unthinkable. Don't stop till you come first. Life's too unpredictable. Take control of what you're worth. Wasting time's forgiven.